I sit within entomology and I look after a collection of 2.5 million insects. That keeps me busy. So this is the collection centre up here, and within the building we have kind of specially designed storage to house our very important collections, and these collections are visited by usually 150 or so visitors each year. Obviously we don't have visitors accessing the collection at the moment, but hope to return to that potentially next year. So that's me. I am assistant curator of entomology here, and it's a very dynamic role. So I'm out doing field work, processing specimens, identifying specimens, hosting researchers and visitors to the collection and making it as accessible as possible. But obviously I've specialised, and I specialised in carrion beetles because of my undergrad actually. So I did forensic science as my BSc. I thought I wanted to maybe be a fingerprint expert, but then I was introduced to entomology, in particular forensic entomology, and how insects can be used to solve crime. And this just really captivated me. I became quite obsessed with this topic, and I conducted my final year project on this. So I left out a lot of rotting rabbits and monitored the insect succession on those. So I thought this is what I wanted to do. But then I went on to do a master's in entomology and realised that there's actually so many different insects out there. I might broaden my horizons. I applied for several jobs and ended up in Edinburgh at the university, and I was doing a job working on burying beetles. You can see from our beautifully decorated lab door here. So this was just working with one species, which we'll come to a bit later on. Nicrophorus vespeloides. This is our most common burying beetle. And this was, my task really was to breed huge numbers of these beetles. So within every box here, the larger ones contain families, so 2 parents and a dead mouse, and the smaller boxes are individuals. So at any one time I had about 3,000 beetles to feed and to design experiments with, which was brilliant. That was just one species, so I ended up at the museum because I wanted to be working with a whole diversity of insects. And at the museum, I am able to specialise, so I do a small amount of research. Obviously, my focus is mostly British beetles, in particular the carrion beetles, but I am beginning to develop a broader knowledge of the world Silphidae as well. And particular interest of mine at the moment is the larvae, and we don't currently have a key for the British carrion beetle larvae, so that will be something to work on in the future. And one of my favourite parts is learning from other experts. So I've had the real pleasure to study with Jan Ruzicka and meet Derek Sykes, who are 2 world authorities on carrion beetles. And you may have heard of our recording scheme. So because I can't get enough of beetles in my work time, I devote some of my spare time to working on this voluntary project. So it's not just me. I work with 2 colleagues on this, Matthew Esch and Richard Wright. These are both 2 other coleopterists who, sorry, I'm in work, so I may get interrupted at some point. It all just gone. So yeah, Matt was doing research at Edge University on sylphids, so that's how we got connected. And Richard Wright is a well-known British coleopterist, and he'd already written some fantastic identification keys and had already actually had the idea to come up with this scheme, but we relaunched it in 2016. So what we do is we dedicate our time to voluntarily verifying records on iRecord, trying to inspire more recorders like yourselves today, and hosting identification workshops and raising awareness through social media. So it's been a great pleasure working with Matt and Richard over the last few years, and really the scheme wouldn't exist without their help, so it's definitely a team effort today. I'm going to go back to basics just in case there's anybody here that is really brand new to beetles. I've seen a couple of names in there, really nice familiar names, so hello. Beetles. Well, what makes a beetle? Obviously, it's got the typical insect body plan of having a head, a thorax and an abdomen, and of course 6 legs. For beetles, the diagnostic features are that they have the hardened forewings, which we refer to as elytra. So this is like the shell-like structure, and it's one of the features that help make beetles so fantastically successful. They also have chewing mouthparts, so they have mandibles, which you can just about make out on this picture here. But particularly for carrion beetles, to recognise them, you either recognise one subfamily or the other. We'll get to this shortly. But overall, these beetles are quite large and robust. It's what makes them kind of what I call gateway beetles. So they're quite easy to recognise beetles in the field. So they're a nice introductory group to hopefully inspire you to become interested in all sorts of different beetles. So they're quite large. They have 10 or 11 segmented antennae, and some have very distinctive clubs, like these burying beetles. They're usually dark in colour, so several of them are black or reddish, and then we have several that have orange markings across the black wing cases. They have truncated wing cases for these burying beetles as well, so you can see the tip of their abdomen poking out, and that also happens with some of the Silphines as well. And if you want to get into the detail, the tarsal formation is 5-5-5, which means that each tarsal, each leg is comprised of 5 segments here on each of those. But really, once you've recognised a couple of sylphids and get your eye in there, you'll be able to do them without counting tarsal segments. So carrion beetles, most of them are necrophagous. So this means that they feed directly on the carrion. They are attracted to carrion vertebrate carcasses. And they have a couple of other common names. So what I do is I tend to refer to the whole family, Silphidae, as carrion beetles. It should be carrion beetles and their allies, because there are a couple in the group that aren't carrion feeding at all. But overall, most of them are attracted to feed and breed on carrion. Their other common names for part of the group, the Nicrophorus, are burying beetles or sexton beetles, and I'm going to explain why. But yeah, it's important to recognise that some of them are actually herbivorous, and we have a couple of predators in the group too. So this is what draws me to sylphids, because they're such a small group. We actually just have 21 species recorded in the UK, but they are incredibly eclectic and a dynamic little group. So we have the carrion feeders, but we have the predators, and some of them are quite attractive species too. So as well as having forensic applications, which makes them pretty useful beetles, the most important thing about these guys is that they're decomposers. So what they're doing in nature is they're helping to break down this nasty waste matter, this organic waste, and they're recycling it either by creating new life, but they're also breaking down this waste so to reduce all the nasty bacteria and flies as well that we could have pools and rivers of if we didn't have any of our decomposers. Now, the most fascinating thing about burying beetles is that they have this unique behaviour. So you may have heard of this already, but we call it biparental care. It's because both the male and female care for the young. And it's a little similar to birds raising their young in a nest. You can think of it like that. But this is incredibly rare amongst insects because they are non-social insects. So what happens is the male and female will locate, or a male or female individually will locate a small corpse. This is usually a small rodent or a bird, perhaps, but they will approach larger carrion too, such as deer, foxes, badgers. And what they'll do is, if the male reaches the carcass first, he will signal for a female. So he will assume a posture where he sticks his abdomen in the air, and he will release pheromones to try and attract a mate. And then what they do is they work together to bury this resource. Oh, skipping ahead. So they're burying it to avoid competition from other scavengers, because obviously there are lots of flies and other beetles and foxes, badgers that would be interested in this resource. So what they do is they do literally bury it. They'll excavate soil from underneath the corpse using their legs and their very broad heads, and eventually the corpse will sink down and they will cover it over. They then begin to prepare the carcass. So I hope nobody's just had their breakfast. What happens? I think you know what you signed up for today anyway, talking about carrion. they will prepare it. So they will strip the carcass of any fur or feathers. They then kind of shape it into a ball, and they will cover the carcass in anal secretions. So these secretions contain antibacterial properties, which actually help slow down the decomposition and preserve this resource for longer, which is incredible. So they're doing this, they're maintaining the carcass, and in the meantime, obviously the male and female have mated, and she has laid eggs in the soil nearby. And once these hatch, they, you can see this sort of ball-like shape of the carcass here. The larvae, once they hatch, they enter the top of the nest where the parents have made a crater, so they've chewed a small hole in the top there, and then they are very lovingly fed by the parents. So both male and female will feed on the flesh of the carcass and regurgitate to the young. It's just so sweet, really charming family setup. But they're doing this, they're investing all this care in raising this one brood because of this scarce resource and the competition that they face. So A, they're protected underground, and B, they are directly feeding the offspring to make sure that they are getting enough food and developing correctly. But there's all sorts of drama that happens within the burying beetles family. So unfortunately, if you're a larva and you beg too much, you may be killed by your parents. And when I say begging, the offspring aren't able to vocalize, so their form of begging is tactile. And what they do is they rear up and they, with their legs, they'll touch the parents on the face as a request for more food. So the research on that was by Claire and Per Smith at Edinburgh University, and there's all sorts of other incredible kind of dramas, and it's almost like a soap opera going on within the beetle family. You have rival males coming in to try and take over and kill the dominant male. You have the same happening with females. It's all very dramatic. So yeah, the burying beetle life history makes them really, really fascinating. What's more is that although the offspring don't communicate vocally, the parents do stridulate, so they are able to produce noise in a similar way to Orthoptera, and they have what we call a stridulatory file. So if you can see just in the center of the abdomen here, there's these two little lines. This is the file, and then on the tip of the elytra on the underside, you can just make out there's some hairs here. So these little bristles are passed across this file, and it produces a very squeaky noise, and this is where the beetle will be just moving its abdomen in and out, scraping past the wing cases there. So unfortunately I did have a little video clip for you, but I don't think the sound is going to work for us here, so I'm just going to switch my screen and we'll just look at it through Twitter instead. Hopefully you're going to be able to hear this okay. Perhaps not. Craig, can you let me know if that is audible?
You need to share your sound, Ashleigh, when you share your screen. There's the little switch that you...
Yeah, I was just hoping it would come up at the top again, but I'm going to have to reshare. Sorry about that. How about that?
Yeah, we can make that out, but it's very, very quiet.
That's a shame. What I'll do is I'll also just pop this link in the chat. This video was taken by my friend John, who I worked at Edinburgh University with in the Beetle Lab. So thank you very much to him for sharing this one with me. I think if you can't hear it now, I would definitely recommend having a listen later on. Very, very squeaky. So if you do disturb carrion and you hear a squeak, yeah, that's the beetles. You'll sometimes see them before you hear them. Sorry, just reshare that. Okay, sorry about that. And when you find carrion beetles, especially the burying beetles, they are sometimes covered in an enormous number of phoretic mites. Now, these mites are generally considered to not be harmful to the beetles. It's deemed to be a kind of beneficial relationship that they have, whereby these mites are interested in feeding on fly eggs and larvae. So they will hitch a ride on the beetle. I refer to this often as the beetle bus, and they will then head with the beetle to the carrion, and then they will hop off at the Carrion bus stop, and they will then predate on the fly eggs and larvae that are there. So for the beetle, this is great news because these mites are helping eradicate the competition. So fantastic mutual relationship there. But there has been some research that has shown that actually these mites will also occasionally feed on the eggs of the beetles too. So it's a bit of a grey area. There is still research going on into this, but something worth considering. Generally, if you see a beetle overloaded with lots of mites, it's fine. They manage quite well with them. I have seen several that have been so overloaded that I'm quite sure it probably starts to affect their fitness and would make it difficult for the beetles to fly. But that's me just speculating there. It's important to recognise that it's not just the burying beetles, it is some of the non-burying beetles too, but usually those that will be associated with carrion and potentially dung as well. So there is very, very high competition. You'll see if you start doing any carrion trapping, or if you now just are more intrigued and you will check any carrion or roadkill that you find, there will be lots of flies, lots of different types of flies and beetles that will be feeding on the corpse. So it's really, and this is how this behaviour has evolved with the burying, that they really need to try to fight against the competition. So you probably got the link at the very beginning that I dropped in the chat for you. This is a link to our recording scheme homepage on the UK Beetle Recording website. So very importantly, there is a free identification key on there for you. So you can download that one and use that one at your leisure. Do have a look at it after today's session just to familiarise yourself with the layout. It is a working copy. This was originally produced by Richard Wright, and Matt and I have since contributed and we've modified it in certain ways. But if you have any feedback on that key, please do let us know. Our email address is on that web page too, as is links to iRecord and also our Twitter account. So we are quite active on social media, and it's one of the best methods of getting in touch with us. We don't check the scheme email as regularly, but we are pretty responsive on Twitter. So you can always get in touch with us that way if you've got any ID queries or questions, or potentially some fun photos to share with us. So please go ahead and follow us on Twitter. We've also got a little link to this little photo that we put together just to help if you're a moth trapper. So carrion beetles some of them are attracted to light, and this is a nice, clean, non-messy way of recording some of them. So they are attracted to light traps. They can cause havoc in the light traps and feed on some of the moths in there. So sorry about that if you're on team moth. But there are just a few of them, and this is also available to download on our webpage if you want a copy. At that point, I'd just like to pause and maybe take any questions from you. Yeah, so potentially Craig can read those out if you have any, or I can quickly just stop sharing so that I can see you, because I can't see comments in this setup right now.
There's no questions in the chat at the moment, Ashleigh. I just reshared the link to the ID key that you shared earlier, because a few people joined after you'd shared that. So that should be available now, the coleoptera.org.uk link that I've just reposted.
That's great, thank you.
Craig McAdams got his hand up. Can you unmute yourself, Craig?
Yeah, sure. Hi, Ashleigh.
Hiya.
I was interested in the stridulation. Can you identify the species by the stridulation? Do they have different songs for each species?
Yeah, that's a really good question. There's a couple of papers that have looked into this, and potentially some research going on at the moment, so I won't say too much, but yeah, it has been suggested that that is the case. There's certainly different shapes, so we call it the stridulatory file, or the pars stridens, and if you compare species, these are slightly different shapes. So potentially producing a different sound. But yeah, there is some research into this in progress. And I should also mention, I completely forgot, that the stridulation is much more complex than just simple defensive or what I initially thought it was anyway. So when you disturb these beetles, they will stridulate, they will make the noise. So that is a defensive mechanism there. But they have 3 types of stridulation which have been documented. So they will do the defensive one to ward off predators, but they also have the mating stridulation to attract a mate or just to communicate with a mate. But they have the call to the young as well. So they have this I've forgotten the name of it now. This other form of stridulation, which is basically signaling to the offspring it's time to feed. Because once the offspring get to a certain stage, some of them are able to self-feed, so they may be wandering around the carcass on their own, but the female can and the male can call the young for direct feeding. So yeah, the stridulation is quite complex, and there's definitely a lot more research to be done in that area.
That's fascinating, thanks.
We do have a few questions coming in now, Ashleigh.
Sure.
Firstly, from Edward, when taking a photo for submission to iRecord, what features of the beetle do we have to make sure are in focus or in the shot?
That is a brilliant question. I'm so glad you asked, but I'm going to cover that later on. Basically, you want a dorsal shot. I'll show you some images later. Dorsal shot that shows you the antenna, ideally the thorax and the elytra as well. Now, my biggest tip would just be to take several images, because it's, as I'm sure you know, it's quite tricky sometimes to get all of these features into one shot and everything be in focus, unless you're stacking it and creating some masterpiece. So as you can see from this image that I've got up at the moment, we've got the antenna visible, the thorax is visible, we'd need to see that if there's any hairs, and the elytra is going out of focus slightly at the bottom. Now, we will come to this one later on, but we do sometimes need to see the legs and also the abdominal segment. So if you can just take multiple photos from slightly different angles, but a dorsal shot is the most useful for us.
Excellent. That's good to know, particularly for all those on Team Moth.
Yeah.
Another question from Anne-Marie this time. How do they actually strip the fur from the animal?
That's great. They've got fantastic mandibles, and they kind of shave the fur off. So they'll chomp along, and they get very, very close to the skin surface. So they do a very, very good job of it. But it's those strong mandibles.
Okay. And is there a particular season when they appear in the field in larger numbers, in a bigger population? So what particular season are you best to look for them?
Yeah, good question. So they overwinter, so you can't generally find the Nicrophorus species or many of the Silphine species at present, but they emerge from sort of April onwards. Some we have had records from March, and they will be active throughout the summer months, but to kind of avoid competition from other species, it is slightly staggered. So you have a couple that are more summer breeders and some that are spring breeders. You'll notice a big emergence of Nicrophorus humator coming out from overwintering in the early spring. So this is the all black one, which we'll get to shortly. So for moth trappers, you'll notice a real big boom of that population. It is our most common burying beetle that moth traps. And yeah, so it varies across the species. Did that answer your question? I hope it did.
Yeah, I think so. I mean, for a diverse family, you're going to have different seasons for different species, aren't you?
Yeah.
Another question from Richard this time. Is the terrible smell I associate with these beetles a defense mechanism or from the anal gland secretions?
Yeah, it is a bit of both. These secretions don't smell very nice. I've kind of got used to it, so it doesn't bother me so much, but I know for moth trappers it can be quite unpleasant. And if you put them in a pot, it can ruin that pot for life. It comes from if they've freshly been in carrion, they can be quite coated in it and quite stinky that way, but it is also from the anal glands and those secretions too. But this is very, what's the word? It's very subjective, because I have obviously working in a lab, there were several of us working on these beetles, and to some of us they smelled quite vile, some kind of passive, but others thought that the burying beetle larvae smelled like peanut butter. So yeah, up to you what you make of it.
That's not a bad smell.
I didn't think it was peanut butter.
That's great. Thanks, Ashleigh. I've just got one quick question about the mites, the parasitic mites. Are they larvae when they're on the beet? Are they parasitizing as larvae, or parasitizing as larvae and adults?
Yeah, they're not really parasitizing. The ones that hitch a lift on the beetle are at the adult stage, and then they'll breed and have offspring when they're at the carrion.
Okay.
So yeah, they're hitching a lift as adults.
Okay, my apologies.
Rather
than parasitizing, right?
Yeah.
Cool.
Right, I will continue if that's all right with everybody.
Yep, crack on.
Good questions. So, identification. Now, I mentioned that there's these two subfamilies, and they look quite different to one another. These are the Nicrophorinae and the Silphinae. So you have your burying beetles in the Nicrophorinae, and they can be recognised by their fantastic clubbed antennae. So it's the sensors on these clubbed antennae that help detect the sulfur compounds released during decomposition, and how the beetles locate their resource. So they have a brilliant club, nice and strong, sometimes coloured orange, so nice and recognisable, whereas the Silphines have very thread-like antennae, and sometimes they gradually expand at the tip, but they never have a really distinct club. So that's the main way to separate the two. So we'll first look at the Nicrophorinae, so the Nicrophorus. We just have one genus in this subfamily. And as I said, our most common of these is the all-black species, Nicrophorus humator. Now, it's worth mentioning that we have had a second all-black species recorded in the UK, that's Nicrophorus germanicus, but its status is a bit of a grey area. We think it potentially was never native and is almost definitely extinct from the UK. So not one really that you're likely to encounter, but for information, Nicrophorus germanicus has all black antennae, whereas humator has orange antennal clubs. So this one is, very common and widespread in the UK, and it's active between February and November. Now, to separate the others, they all have orange markings, and at first glance can be quite tricky to separate. I have it in this kind of table format, which I find quite helpful. So the first one we can get rid of is this one, or easily identify, not get rid of. We don't want to get rid of any of them. So this one is Nicrophorus vespuloides, and it has completely black clubs. So you can see that all the others have orange clubs on their antenna. So if you have a orange and black Nicrophorus burying beetle, and it has black clubs, then you definitely have vespuloides. It's a very smart looking beetle. It's also worth noting that it's not hairy at all, so it doesn't have any hairs on the thorax here. And sometimes if you can only get a photo of the beetle with its head completely in carrion or just covered up in some way, and you can't see those antennae and you can't get them in a photo, we can also confirm this species based on the markings at the back here. And the hairs on its abdomen. So vespuloides often has like two splodges in the second banding. Next we have investigator. This one is incredibly scarce in the UK. You're unlikely to encounter this one unless you're in the kind of Norfolk area. It has golden pubescence all around the thorax, which helps separate it from vespilo. The other key issue here was the hind tibia, so the hind legs, and I like to sometimes call this one, Nicrophorus vespilo, as the John Wayne cowboy burying beetle, because it has these bent legs. Really, really useful to separate these two. Worth noting as well that the hairs can abrade on older individuals, so the legs are the most reliable feature for separating out Vespillo. But if you get a lovely fresh specimen, this margin of golden hairs just along the front there can be really nice looking. You can also see nicely in this photo that the elytra are really hairy, and that's not the case for any of our other British species. Then we have investigator. This is another one that's very, very common in light traps. We've also called it the banded burying beetle. It has those orange clubs. It has a hairless pronotum. And then the other thing that you need to look at for this species is the banding. So the banding on Nicrophorus investigator is often not interrupted. It's continuous across the suture, but this is a variable feature. So the other thing you need to look at for this species is the hairs on the abdominal segments. So there should only be golden hairs on that final segment there, and the rest you can see are black. Now this can be easily confused with the neighbouring species here, Nicrophorus interruptus. And this one has, again, the orange clubs, hairless pronotum, but in the old keys, it would say that this the main diagnostic feature was that this band here was broadly interrupted. But of course, we've just seen from the other specimen that that's not always the case. Like there's a bit of confusion there. So the main difference is that look at this. There has got lots of golden hairs on the abdominal segments. So it's really key if you've got one of these two to get a nice image of the bum of the beetle. There we go, so you can see broadly separated here, but most importantly, golden hairs on that abdominal segment. So we're just going to have some, just pick you on the spot a little bit, have a little practice, see how you've all been listening. And I've just got a couple of photos to see if any of you are willing to be brave enough and suggest a species for them. So it's up to you how we do this. You can drop your suggestions in the comments, or if any of you want to raise a hand and turn your mic on, you're welcome to suggest. So I'll let Craig manage that bit as I can't see the chat. First up is this one here. So take it in, have a look at those different features, thorax, the elytra, the antenna, and have a think about what this one could be.
Yeah, a few people are commenting in the conversation, Vespiloides.
Excellent, fantastic. Yes, this is Vespiloides, so our common burying beetle, and it has those black antennal clubs, so a really nice simple one for you. And this is a fantastic photo from Liam Olds, and you can see those mites that are hitchhiking on there. Well done, folks. We have another one here, a little bit trickier because only part of the beetle is in focus. So what do you make of this one?
Oh. Yeah, I've guessed Vespillo, but a couple of other people are saying investigator.
Sorry, Craig, the others are right.
Oh.
It is investigator, yeah. So I've been quite cruel. It's a photo I took, so it's pretty rubbish. Not much of it's in focus. The legs that you can just about make out are straight, these hind tibia. You've got the top banding, which is continuous, it's not interrupted, and you can just make out that we've got golden hairs on the final segment, but it looks like darker hairs here. The other fun feature about investigator, please don't use this as a solid identification feature, but it has two Scotty dogs on its elytra. Once you see them, you can't unsee them. So that's another little tip there. But again, don't just rely on that. Okay, so now we'll hop over onto the other subfamily. So this is the Silphinae. And again, I mentioned that they have these thread-like antennae. They don't have a really distinctive club. So that's how we'll recognize them. Now, the biggest beetle in the Silphinae is Necrodes littoralis, and this is sometimes called the shore sexton beetle. It's a big beast. In fact, might be a good time to just get my box out so that you can see the different sizes of these. You see, I'm in the museum today, so we have access to lots of specimens. But this box here contains examples of all the British species. So size-wise, burying beetles here you can see are generally much larger than the sylphines, and this is Necrodes littoralis, so it is generally much larger than the other sylphines. It's all black. It has a thread-like antenna, but on a live specimen you'll notice that the final three segments on the antenna are almost bright orange. Obviously this photograph was taken of a preserved specimen, so it's not as vibrant, but that is the way to separate this one out from some of the others. Its size, its orbicular pronotum, it's very, very round here. It's hairless, a chunky beast, and then it has the thread-like antenna with the golden orangey coloured tips. It is attracted to light like the burying beetles, but it's really the main one of the sylphines that is. So your moth trapper, this is the one of the sylphines that you're likely to see. Mostly active between May and October, and it peaks in the summer in August, and it's very common and widespread. It gets the name littoralis because originally recorders thought it was associated with the shore, but we have many inland records, so don't assume that this one is coastal. Occasionally people do muddle this one up with Nicrophorus humator because they both have the orange tips to the antenna. But remember that these don't have distinct clubs, and once you have them side by side you can see how different they are. Necrodes has generally got like a much flatter appearance, and it also has these hunky if you're a female maybe, these chunky hind femora. So these chunky thighs on the male only, they're not present on the female, and these are believed to be used in copulation. Then we have the predators. I mentioned that some are not carrion feeding, so we have 2 or 3 predators here for you. We have Phosphuga atrata, which actually has a subspecies, subrotundata, and we won't worry about that one for the purpose of today. We'll just focus on these 2, and they are both snail feeders. And they have narrow heads which enable them to access the mollusk in the shell. So I absolutely love this picture here from Maria. She's caught the beetle in action. So the common snail hunter, it comes in 2 colour forms, black and red, and they can often be found together. And the life history of this one is the beetle, it's quite a badass predator. It will bite the foot of the mollusk so that the mollusk retreats into the shell. It then administers digestive enzymes which start to break down and break through the mucus that the snail produces, and starts to break down the flesh of the poor animal. And it can then really just get right stuck in there with its head and devour the mollusk. So they're excellent predators. Not so good if you're a snail. They're active between April and September, but actually now is the best time of year to find them. So if you check under any fallen logs or under the bark of dead wood, you will find some of these beetles. They also can be found under moss, just generally in nice cozy places where they would be overwintering. Sometimes they're found in clusters, sometimes they're on their own, but definitely easier to find them now than in the summer months. If you are looking under bark and things, though, please remember to try not to destroy too much of the habitat and put the bark back when you have finished taking photos, of course. Then we have Ablateria laevigata, which we've called the smooth snail hunter. For obvious reasons, its elytra are completely smooth, so it doesn't have any of the ridges on the wing cases that the other beetle, Phosphuga atrata, has. Very, very smooth, and it has a different shape to the pronotum, which you can see as well. And that one's active between April and September. And then this one is probably my favourite of the predators, Dendroxena quadrimaculata, and it's a very distinctive beetle. There's actually no other British beetles that you could get this one modelled up with. So it has this lovely orangey brown colour to most of the wing cases, and then it has 4 spots of black there. Now this one is a predator of caterpillars, moth caterpillars, found on oak trees. It can also be found on hawthorn and a couple of other bushes and shrubs too. It's naturally scarce, and it is found April through to June. We don't have that many records of this species, so it may actually potentially be more active than that, but the more records we get, the more we will know. In Scotland, we just have a couple of pockets for this species. I'd be very, very interested if you're able to go out and bash some oak trees for us to potentially find more records of this one. Yeah, fascinating little beetle. Then we have Oiceoptoma thoracicum, also known as the red-breasted carrion beetle, and this is extremely common and widespread in the UK. It does prefer woodlands, so if you're out on a woodland walk, this is one that is day active and you'll sometimes see bumbling along paths, but particularly on any dung, carrion or fungi. It has a particular fondness for the stinkhorn fungus here, as you can see, and yes, it will also go on dung too. Active between April and August, and a woodland specialist. And we have the other Silpha species. Now, at first glance, these can look pretty similar, very aware of that. But once you get your eye in, you'll realise that there is some key differences here. So one that I like to mention is, we haven't got it in the key, but when you compare them side by side, Tristis has almost a rectangular appearance, whereas the other two are a bit more spherical. The main difference here is having a proper look at the punctuation on the elytra. So when we zoom in a bit, which you can do with a hand lens very easily, you can see the different textures on the elytra here. So obscura is very matte and it has very regular uniform punctures. We have Tyrolensis, which is more, it's got like a wrinkled look to the elytra here with irregular punctures, and it's got a shine to it. And then Tristis is also matte in appearance. We go back, you can see it's quite matte here, but the ridges on the elytra are shining, so that's a helpful thing. And in the key we do put as well that if you've got a specimen that you've retained and you look at it under a microscope, you'll see that it has these shining tubercles. So for every pit that there is, there's a small little bump that's shining. This is Silpha tristis. I will say as well that tristis is sometimes confused for Phosphuga atrata. So we flip back here, because it has these shining ridges, but atrata is very, very shiny, whereas tristis is very matte and it has a different shape to it too. And don't forget, it doesn't have the skinny head that the snail hunter has. I hope that one's clear for you. If you have any questions though, pop them in chat and I can answer them in the next break. And we have Silpha carinata. I just wanted to mention this one because this is a real kind of favourite of mine. It's very, very scarce, potentially extinct in the UK. It hasn't been recorded since 1985, and it was known from the Wiltshire, Oxfordshire area, in particular this one site called Great Ridge Wood, which I visited with Mark Arbuckle and Matt Esch from the scheme a couple of years ago now to try and survey at this place where it was found by Nash previously. We weren't successful that time, but we're going to try again. The carrot feeder, this indented anterior, and yeah, hopefully we will find this one again at some point. If you're in that area, let me know and we can chat about potential trapping ideas. Then we have 3 species of Thanatophilus. My favourite of these is Thanatophilus rugosus. It has this wonderful wrinkled appearance, and we have actually assigned it a new common name, the wrinkled death lover. It's a good one. So that one's really easy to separate from the 2 other Thanatophilus species. It's also the one you're most likely to see in Scotland. Thanatophilus sinuatus, it has the same body shape, but if you notice, it doesn't have any of the wrinkling on the elytra. It's very, very smooth. We've called this one the smooth death lover. And a feature of this is that it has a very tiny and sometimes difficult to see shoulder tooth. So we're going to have a look at specimens under the microscope after this. So we'll see if we can show that one to you more clearly then. And the 3rd species of Thanatophilus is dispar, and this comes in a black form as well as the red form, so it looks quite similar to sinuatus, but the difference here is that it has hairs on its elytra. So that's an important feature, and it doesn't have a little pointy tooth on the shoulder of the elytra. It's also incredibly scarce. So in Scotland, it has strongholds on Uist and at Loch Leven, but it is lost from any other former sites in England. It does have a stronghold in Ireland still, but really this is a real scarce beetle. And we have Aclypea opaca, which I think looks almost like the teddy bear of the Silphids. It's got this dense golden pubescence that covers the head, the thorax, and the elytra. So can't be confused with any of the others. It does have a sort of similar body plan to Thanatophilus, but when you look closely, you can see that the anterior margin of the pronotum is almost curved outwards, whereas Thanatophilus is indented. Yeah, it has this beautiful pubescence, which if you catch it in the sun is very shimmery. It's a vegetarian, so it does feed on beet, which is how it got its common name, the beet carrion beetle. It's not a carrion beetle at all. Yeah, definitely vegetarian. So both the adults and the larvae will feed on the leaves of these plants. It is nationally scarce. I have found it in Scotland, but it is also found in the kind of Norfolk, Suffolk area. And we're not going to cover the identification of Silphid larvae today because that's pretty complex, but it's worth just mentioning if you do think you come across any, be seriously interested in any photos and potentially any specimens that you retain. You can easily separate the two subfamilies, like you can with the adults, based on their exterior. So you've got the very fleshy coloured larvae of Nicrophorus. So they don't need to be as robust as the sylphines because remember they're underground, so they're protected. The parents are looking after them and they are concealed from any predators. But the sylphines are much more skeletorized. They are sometimes confused with woodlice or carabid larvae. So they have this tougher exoskeleton because they're out and about. A number of them are predatory, but they'll also be feeding on exposed carrion. So they are tricky to identify. There are some great partial keys out there. So Jan Rzyska provides keys to the Nicrophorus, and his group at the University in Prague are working on keys for some of the other species too, particularly the Thanatophilus. So they are available, but we don't yet have a fully formed one for all the British species. So please get in touch if you think you find Silphid larvae. Definitely worth taking photos, but bear in mind that we probably can't identify them from photos and that we do need voucher specimens. So here we'll just have another quick practice. I've just got one for you from this section. So if you'd like to just have a little gander at this one and see what you think it is. So remember to look at those antennae. Look at the kind of texture here of the elytra.
A couple of people are going rugosus.
Fantastic. Yes, excellent. It is indeed rugosus. So a really nice, easy one to spot, I think, because it has this wrinkled, lumpy, bumpy elytra. Great stuff. So on to trapping methods. They are opportunistic beetles. So carrion is a limited resource, and there's lots of competition for it. So they have to be opportunistic. They're obviously using those incredibly powerful antennae to detect the scents released during decomposition. So they will go to wherever the carrion is within reason. Obviously, they do have some habitat preferences, like some are woodland specialists, some prefer open sites, some prefer different soil types, but these beetles are able to move, the burying beetles at least, are able to move the carcasses a small distance if the soil type isn't quite right. But they're not going to move it across counties or anything. So, but they're strong flyers. They are capable of flying good distances. I should also mention that they're actually mostly diurnal or nocturnal. They're nocturnal or what's the word when they come out at dusk? Forgotten it. Sorry.
Crepuscular.
Crepuscular.
Thank you, Greg. Thank you very much. Yeah, crepuscular. So most of them will just be seen at night, which is hence why they come to light traps. But Vespuloides is a day flyer, and you'll occasionally get a couple of the others flying during the day too. So if you want to find them, the best thing to do is to create a trap. You can check any roadkill, any carrion that you find. I would just like to say, please be a bit safer than me. Obviously, I'm on a road here. I'm not wearing any gloves. I would recommend using gloves if you are going to be handling any carrion. So best way to do it is to set traps. You can run a light trap, which I mentioned is the easiest non-messy way. And that will attract those 5 or 6 species that we have attracted to light, but you'll be missing those other ones and really interesting beetles. So I would recommend doing baited traps. And there are a couple of different ways of doing this. And when I say bait, there's a whole host of different baits you can use. If you want to add some realism to it, you can provide mice bait. You can purchase these from a pet store. And the tip that Matt and I often give is that it's really important to mature your bait. And when I say mature, it's because the beetles tend to find the carrion a little bit later than flies do. Takes them a little bit longer to locate the carrion, and potentially they're also using a cue that comes in at a slightly later stage of decomposition. So if you mature your bait, let it ripen up a bit, that will help with attracting the beetles. And when I say mature it, just stick it in an airtight container, leave it in a warm place where people don't mind. So that's one method, but you can also just use rotten fish, which is really quite pungent. Anything, any off meat really. And you can either stick it in a trap, or you can also bury it. And for one of our workshops, we buried 3 hare carcasses, and we then exhumed them for the group to search for beetles and then identify their catch. And this was fantastic. And it was actually the best way of finding larvae as well. So if you're hoping to catch any larvae, burying them is a good method for trapping. Obviously, you have to have access to a larger carcass there, and also we didn't do it this time, but really to avoid the interference from scavengers, you should also have a kind of protective cage over your carcass, because you will get larger scavengers like foxes and things that will try to take away your lovely bait. Which is why sometimes having hanging traps is beneficial. A hanging trap can be as simple as a yogurt pot with the bait in, hanging up in a branch. But if you want to potentially, be able to retrieve your beetles and take nice photos, you want to keep your bait separate from where the beetles will come into. So you want to have it in the trap to attract them in, but you don't necessarily want them running around in it and getting coated in the decomposing stink. So you can have your bait hanging. And this is a version of a pitfall trap. So here, this is Matt's design of having the carcass dangling above a pitfall trap. And you can do a similar thing, but in a hanging trap as well. So this means that the beetles are attracted in, but they will generally be crawling along here and fall into the trap. If you're not familiar with a pitfall trap, it's as simple as a cup placed into the ground. And the most important thing is that it's in completely level with the ground surface, so that the beetles don't have to go up into the trap. They're just walking along and drop straight in. And then really what I would recommend for pitfall trapping and hanging trapping is that you check these daily if possible, or at least every other day if you're doing live trapping, and put some substrate in the bottom, a bit of sand, soil, leaves, debris, so that the beetles have a little bit of cover from one another, because they will predate on each other, and you won't have any beetles left to identify. So again, regular inspection of traps. But if you are surveying or wanting to retain voucher specimens, you can use a preservative in the trap as well and leave them for slightly longer. Then recording your beetles, because this is what it's all about. Really, it's a plea from us to please record your sightings of carrion beetles. We recommend using iRecord. It's a fantastic website and app. It's really easy to submit your data. I'm sure many of you are already very familiar with it, and I will also be promoting it too, but it's really nice to use, and most importantly, it reaches the national recorders. So many verifiers on there are from recording schemes, national recording schemes, so they're able to see your records, verify them, get in touch with you for any additional information, and then really importantly, all this data gets passed on to the National Biodiversity Network and updates the distribution maps for our British species. And then this data is accessible and can be used by a whole host of different scientists, so you can actually support research and even decision making at both local and national levels. So really important, and definitely something to invest a little bit of time in doing. I will apologise in advance that we do have a backlog of records to verify on iRecord. We're doing this in our spare time, so bear with us. But if you ever have any records that are sitting on iRecord for a while and we haven't got to them yet, feel free to just get in touch and drop us an email and we can take a look at those records for you. Again, with any issues with identification, just get in touch with us. So there is the contact details on our scheme page, but you're also welcome to email me at the museum too. This is a format for iRecord. Very, very easy to submit records. You can enter a casual record, or you can also upload a spreadsheet of records. There's the form there. And somebody mentioned about record shots. So that was a really great question, and hopefully this covers it a bit more visually for you. We don't need a scale, it's a bonus. So main thing to do is just get a nice dorsal shot that features all the relevant components of the antenna, the thorax, the elytra, and ideally those back legs and hairs on abdominal segments too. So that's a really great one there from Glenn. We have another one here, and I use this one to say, please don't be worried about submitting blurry photos to us. I would much rather have a photo. It's a bit blurry than not have a photo at all. It really, really, really helps. So I can see everything I need to from this photo here. It's got the curved hind tibia. It's got incredibly, it's a very hairy beast, and it's got that margin of hairs at the front of the thorax. So I know that this one straight away is Nicrophorus vespillo, bent-legged burying beetle. And then this one from Martin is brilliant because it is absolutely covered in mites. It really, really is. So you can't be sure about the hairs on the pronotum here. You can't see the pattern of the elytra, but what can you see? Maybe I should let people put in the comments here. Let me know if you've got any more. You can see the antenna, yeah, so those are orange, so we know it's not Vespiloides, but what else can you see that might indicate the species of this one?
Curved legs?
Yeah, spot on. Excellent. So this one is Vespillo, but it's really just overloaded with mites. So I'm going to switch over to the microscope now, but whilst I'm getting it set up, I can take a couple of questions too if anyone wants to drop any questions in now. And I'll just stop sharing this screen.
Yeah, a few questions came up while you were talking there, Ashleigh, that I'll go through one at a time. First, Nick had asked about the oak species that you had referred to. I forget which species it was now. Yeah. Is it best found by beating foliage?
Yes.
Yeah. Okay. Georgina had asked, how far can carrion beetles fly or disperse?
That's a good one. I actually looked this up recently because I couldn't find an exact reference that described it in miles, but the Czech reference that I have is several kilometers.
Is that generally as a family rather than individual species?
So that's how far that they can travel to locate carrion. When they disperse, as larvae, they're in this. When they get ready to pupate, they move away from the carcass, which obviously helps with the threat of predation. So they'll disperse in the soil. I don't actually know of anything about how far a family will disperse in terms of distance. Right. But with their ability to fly quite well and the need to search for carrion, reasonable reasonable distance, yeah, I would say.
Edward has said he thinks it's about 6 kilometers. What was it you said? You'd said
Several, yeah.
several kilometers. Yeah, he'd said he thinks it's about 6.
Okay, you can send me that paper, Ed. Thank you.
And Craig McAdam had asked, why are they attracted to light when they are finding the carrion with olfactory cues?
I do not know, Craig. I do not know.
Interesting.
So I'm just going to switch screens for you. Wake the camera back up.
Your just a comment about your slide about the larvae, Ashleigh. That's really interesting. It's helped me narrow down a particular invertebrate that I found a couple of years ago and never knew what it was, but I think it was a Silphid Larvae. I'll send you a picture afterwards.
Oh, fantastic. Yeah, please do. There are a couple that we can do from photographs. So Silpha tristis is a nice, easy one to do, and Oiceoptoma is another one we can do. But yeah, don't be disappointed if we can't do a Silphid larva. Right, I'm just going to, oh no, just need to minimize that one first. Okay. Okay. Live specimens, here we go. How is that for you folks? Can you see my lovely beetle here?
Yeah, we can see that.
So who recognises this one?
Quadrimaculata.
Fantastic, yes. So one of the easiest sylphids to identify. Just going to play with my box here. So we'll tackle the Nicrophorus first. I should have said at the beginning, Craig, that we have 2 hours to play with on this workshop. This bit is a bit of a kind of a free forming session because it can take as long or as short as you like, but I do just have a couple more slides once we finish this. But you can drop in any questions at any point really for this bit. Now, I also gave you photos to identify things from because that's mostly what you'll be doing. We encourage recording of these species live in the field. There isn't a desperate need to retain specimens for most of them because they're so large, easy to identify, easy for us to verify. But there are a couple of smaller species with more subtle characteristics that would be worth taking a voucher of. So this one here is Nicrophorus humator. It is the entirely black species. It has those lovely orange clubs. And one feature I wanted to point out that we haven't talked about yet is the sexing of males and females. So it is possible to sex some of the Nicrophorus based on a couple of things. So males tend to have, like with some other beetle species, beetle families, they have broader foretarsi. So their tarsi here are much broader and they're also hairier than the males. Males also tend to have broader heads than females do. And there's also a difference in the abdominal segments, but it's best shown on Nicrophorus vespuloides. So let me just show you that one now. Have a lovely male and female here together. So I can just show that one to you. So, oh, it's got quite dirty antennal clubs, hasn't it? what's this one? The one with white clubs. They've both got black clubs. Okay, so differences here on those abdominal segments is that the males end in a very small tip of the abdominal segment and the females have a very large one, which is actually two fused together. And there is the difference in the head. So the male, you can see, has a slightly broader head here, female, and he has broader, hairier foretarsi. Focus for you. There you go. Jump in if anybody has any questions.
Patrick's asked, does brood size vary for carrion feeders depending on the size of the carcass?
It absolutely does. Yeah, good question there. So they will tend to lay the right number of eggs depending on the kind of size of the carcass. The female kind of assesses it. So obviously if she's got a fairly small carcass, a small mouse, she'll lay a smaller brood. If there's a really big carcass, they may lay a bigger brood. It's also worth noting that if it is a very, very large carcass, like a deer or a hare or something, that they're not able to bury, you sometimes get communal breeding where several pairs will share that resource. So yeah, that's another thing, but the brood size varies depending on how much resource the offspring have available, how much resource the parents have available. Okay, just getting out another one for you. This is another lovely male. Look at those foretarsi, fantastic. So it's got orange clubs. It has that wonderful margin of hairs across the top of the thorax. So who can name this one for us? Dum, dum, dum. It's very hairy. Any suggestions?
Investigator?
Not quite.
Somebody else has gone Vespillo?
Yes, it
Vespillo.
is. Vespillo. So it's got the golden hairs at the front of the pronotum, and it's got those super curved hind tibia. So if I just get you out Investigator so that we can compare that.
So Vespillo is the John Wayne one.
It is, yeah. I hope that helps you with remembering that. Help me. Okay, so here we have a straight hind tibia and a curved one. So if you imagine you were drawing a straight line between the top and the bottom of the tibia here, you would have a huge gap between your line and the middle of the tibia, whereas if you were doing it with this one, it would almost run alongside it. That's a helpful tip. Because obviously if you're in the field, you're not likely to have another one to compare it to. And the Investigator, you can see it has those hairs all around the pronotum. They're not as, it might be a bit braided on the specimen, it's very old, and they're not as dense as the top row, but they are all around, whereas with Vespillo, they are just restricted to the top of the pronotum.
Carrie has a question, Ashleigh. If a food source is buried, do the brood rear through quicker than those in a more exposed setting?
Hi, Carrie. Thanks for joining us. I don't even know. It is possible to find examples where there is a brood above ground. It's not common, so I don't know for certain, but that makes me think that they're not as successful. Obviously, they're just much more vulnerable to predators. So I would say yes, but I would need to double check on references.
And Mike has a question. Can you give any advice on the type of microscope to use?
Oh, for these guys, you don't need anything too fancy. I'm using quite a nice microscope today, but that's just because I'm at the museum. At home, I have a much simpler one. Any stereo microscope will do, really. The magnification doesn't need to be spectacular for these guys. Obviously, what we're looking at is hairs and things. I'll just put another one under. Most of these beetles, you cannot see the features you need to with a hand lens. So you don't need to go to any great expense. But remember that these are gateway beetles, and that once you get the beetle addiction, then you're going to go down that rabbit hole and start looking at lots of other beetle groups, like dung beetles, Kisterids maybe. So worth investing, but I would say you can get quite a good microscope secondhand, and potentially, like, I can send a couple of links round in the follow-up email, Craig, with some resources and things I can suggest to you. If that's helpful.
Yeah, that would be helpful. Thanks, Ashleigh.
So who do we have here? We've got orange antennal clubs. We don't have a very hairy pronotum. We have a gap in the anterior band. And we have golden hairs on the abdominal segments. It's also straight hind legs.
Interruptus.
Yes.
That was Carri, not me.
Carri, you know these. I expect you to know all of them. Oh, look, you can nicely see here that we have. I was going to say male and female. Actually, I think that is another male. Also chunky head. No, this one just has an extremely chunky head. Who is this one? Orange antennal clubs, hairless pronotum, fantastic. It's definitely a male. Look at those tarsi. Continuous anterior band, 2 little Scotty dogs, and we have just golden hairs on the final segment there.
Investigator.
Yes.
Oh, the Scotty dogs gave it away.
As long as you don't all rely on the Scotty dogs. Okay. And then I can also show you, because we're in the museum, I've got access to a specimen of Germanicus. This one is from Germany. It's not a British specimen. Most of the British specimens in collections don't really have data anyway. It's a big beast, so we can't get him all under at once. But you can see he has black clubs on the antenna, and our native Nicrophorus humator has orange clubs. Another feature is that he has the epipleura, which is red. Now this is used in keys, but it's not super reliable for humator. So in Germanicus it's red, you can see. So this area, can you see my cursor okay? So this whole area is called the epipleura, and it's red. But in humator it's usually black, but it can be a reddish colour, as this specimen shows. Oh, sorry, I'm being cat-candid. Yeah, it's a little brownish red. But you're unlikely to encounter Germanicus.
Got a couple more questions here, Ashleigh. One from Lee. Do the Nicrophinae need to have certain soil types to bury their food? Can they bury in clay?
I don't know that it can bury in clay. Yeah, there is a couple of publications on soil type preferences. And as I mentioned, they're able to relocate the carcass if the soil type's not quite right. And I think, very damp clay soils could be quite tricky for them, but they're impressively strong. I'd need to double check on those references, I think, on that.
Okay. And a question from Dave. Hi, Dave. Do these beetles utilize deer growlers or only whole animal carcasses?
Yeah, they will. They will go to those. If you put a trap out that's just kind of like chicken livers or something, they'll come to that. So yeah, they'll come to offal. Whether or not they can breed with it is another question, which I'm not sure about. It might be quite interesting to look into. But they will feed from it.
Okay.
I'm just going to get out some of the predatory species now.
Mo has just commented, even in Sweden, it is uncertain whether we have really had Germanicus. There are notices that it was found during the 19th century, but there is no specimens in the museums.
Yes, very similar for us. It's just a handful of old museum specimens. And it is sometimes difficult to know whether they were genuine, especially when they don't have data. Collectors liked to boast when they had these big, special species. So here are the predators. This is Phosphuga atrata in both its black and its red colour form. You can see its lovely narrow head. So it's very skinny and elongated to help to get into those shells. Thread-like antennae, and you'll see as well that it's very shiny, which is what makes it so different from Silpha tristis. Also, the pronotum is just very semicircular. See, tristis as well is kind of much straighter along the anterior margin. And then our other predator here, Ablateria lobigata, is very, very smooth, which is why you can see we give it the smooth caterpillar, smooth snail hunter common name. It doesn't have those strong ridges on the elytra, and it's not really shiny. It's very matte, but it has that lovely elongate head. I'll just show you that one again as well, caterpillar hunter, just because it's great. Is there any questions on those three?
Yeah, Patrick's asking, can you just remind me what makes these carrion beetles? Is that presumably the snail hunters he's referring to?
Yeah, just taxonomically, they fall into the same family. So you can get within a family very different ecological niches that they occupy, very different life histories. That's just how it goes. So yeah.
Visually is what he was meaning.
Oh, right. Sorry. I thought you meant why they were lumped into the group. So visually, they, very different to the Nicrophorus. The key features are that they have the 11 segmented antenna. They are fairly large as beetles go. The Thanatophilus species have exposed abdominal segment, so the elytra is slightly truncated, and they have the tarsal formation of 555. A bit dark.
Jennifer is asking, when is the best time to go looking for Dendroxena quadrimaculata?
In the spring. So yeah, if you can go April time, keep your eyes peeled April through to June, really. That's when we have records from. Some of our records have suggested that it is nocturnal, so potentially worth going out at night with a torch and checking oaks and hawthorns then.
I'm particularly interested in the Thanatophilus dispar at Loch Leven, Ashleigh.
Yeah,
and
me too.
I'd like to know when that was last recorded, and well, any information that you could give us on when and where to look for that.
Yeah. So we helped RSPB with some of their trap designs and stuff a couple of years ago. So I think the last records are from 2 years ago now. We didn't monitor last year, and we haven't monitored this summer.
Do they come to light?
No.
No.
They so they're associated with lock edges, lock sides, and it traditionally thought that they were utilizing dead fish, but there are records from bird carcasses and I think a hare carcass. So it's really just carrion in that particular habitat. So they like the sandy soils of the lock side. And in the strongholds that are in Ireland, those are around locks as well.
Right.
This has got a lovely reddish colour to it, but as I said, that does come in a darker form. So yeah, RSPB were monitoring at Loch Leven and had some good results there. They found it breeding as well, which is really great news. And it's something that we'll continue to monitor in the future as well. Yeah, I want to go and see it. I haven't seen a live one yet.
Yeah, I've been in this area for a long time, and I've been aware of it vaguely, but I would like to see it now that I've heard a little bit more about it.
Yeah, it was critically. It was a critical species, critically endangered, but it has just been redesignated now. So because of its recent successful populations, it's kind of been downgraded slightly, but it is still a scarce beetle that you know, should have its habitat protected. So
absolutely. There's Jennifer's also asking about Dendroxena, quadrimaculata. Do they come to light?
No, not that we have records of. Oddly, the snail feeding ones we do have records of at light. They're flightless, which is worth pointing out. The records that we have mostly are from the beetles being kind of on the white sheet that a lot of moss recorders will put down by their light traps. So they're not actually in the trap, but of course they are able to climb into some of the traps too, the ones that aren't too slippy on the surface. So that's definitely suggesting that the snail hunters will also feed at night.
Mm-hmm. Kerry has asked what makes an atrata red or black. In some dung beetles, it is probably temperature based.
Kerry, that's a really, really fascinating question, and I do not know. I haven't even read any papers on that yet. Something to look into, because you get mixed. Does this happen with dung beetles too? You get mixed aggregations, like those that overwinter. Obviously, they may not be relatives, but something to look into. The one thing that's really interesting is that Orkney only has the red form, and we're not really sure what that's about. If it is temperature related, then and it's the redder form that you get from lower temperatures, that would make sense because it's further north, but something to look into.
Yeah, she says yes, you get mixed colours in atta and depressus. Interesting about
yeah.
what...
Yeah. The other thing to note is that if you ever find a Nicrophorus humator, the all black species, as a reddish version, that's because it's a freshly or recently emerged specimen. It's still tenural, so its full colour hasn't developed, and that can happen with Necrodes littoralis as well, the big black sulfine. And if you see any of the Nicrophorus with the orange markings that haven't fully developed, they're really funny when they first emerge. They're kind of like a very pale orange, and they just within hours their colour fully develops and they harden up. So now I've just popped those 3 Silpha under the scope here, just so you can have a closer look. So they are quite similar in appearance. This one is very matte. This one more uneven punctures here and very shiny. And then we have this one, which is very matte except for those ridges on the elytra. Anyone want to tell me what this one might be? If you're observant, you'll notice I've got these in the same order as my slide. I should have made it more difficult, shouldn't I?
Atrata from Craig.
No, and I'm glad you said that, Craig, because this is the one it's so commonly confused with. So what I'll do is I'll just pop an atrata next to it and we can see the difference. Oh, there's quite a size difference as well. I don't know if you can see my camera all that well at the moment, but so that's your tristis, Silpha tristis. That's what this is. And this is the atrata. It's a totally different shape. This is a much curvier beetle. Tristis is more sort of rectangular in shape. If you look at the front of the pronotum, tristis is concave. It sort of indents, whereas atrata doesn't. It's nice and curved. Also, the head, hard to see sometimes with Silpha because it's a bit more tucked under, but that just shows you it's not as elongated. So you don't have such a skinny, long head. And then, of course, it's just very, very matte in comparison to atrata. So I hope that one will help you for next time, Craig. Okay, so that one was Silpha tristis on the end, but the other two species of Silpha that we have.
Jane has just commented, or well, asked, how come atrata isn't in the key?
It isn't in the key? Are you sure?
I'm going to go and have a look now as well.
Yes, please do. It was last time I looked. It's Phosphuga atrata. And in the most recent version, we did change that because previously, for many years, it's been referred to as Silpha atrata. But we have aligned ourselves with the nomenclature used on the continent.
Yeah, no, she said sorry. She was looking at Silpha. It's under number 9, elytra
yes.
with raised longitudinal lines.
Yes, so we've confused everybody by calling it Phosphuga now. But it's better that we all speak the same beetle language globally. So, Silpha. We've got either obscura or tyrolensis here. So the difference between them is that if it's obscura, it's very matte, uniform punctures, and tyrolensis is much more irregular, kind of wrinkled texture. So that's what this beastie is. Much more shiny and has that kind of, wrinkled punctuation there. And obscura, very matte, very, very matte. And if we zoom in a little bit, you can see how regular, uniform those punctures are as well.
I have a question here from Nick. Do carrion beetles have an obligatory 1-year life cycle and a single breeding attempt?
That's a good question. Necrodes, Nicrophorus vespiloides will have several generations in a year if it has the opportunity to. Most of the others are single generations. One that's quite interesting to note is Nicrophorus investigator, which is one of the most common ones in moth traps. That's a late summer breeder, so that one will breed at the end of August, and its larvae will overwinter as a pre-pupa, whereas the others which all overwinter as adults, ready for the next year. So that one completes its development the following year. don't know if I've stuck to the original question there. Did I, Craig? Do you need to steer me?
No, that was that answered the question, I think. Yeah, Nick said thanks.
So I just have a couple, and there's actually one beetle that I missed out from the entire presentation. And that is that we have 2 Aclypea.
Sorry, Ashleigh, I'm just going to interrupt for a moment. Yeah. As one or two people have started, one or two people have left, so I'm just going to mention before anybody else does that I've just posted a survey monkey that will only take you less than a minute to fill out. It's just a few questions by way of feedback. If I could ask anybody that's going to leave or before you close your computers, just to take a minute to fill in a few questions, that would be very helpful for us for future events. Okay, sorry, Ashleigh.
No, that's right. And I do just have a couple of slides left just to let you know some exciting things that are happening with the scheme and things, so stay tuned. But just first, I'll just show you the beautiful, in fact, I'm going to take it out of the box here because it's not doing it justice. The beautiful Aclypea opaca, which is just lovely. Look at its golden pubescence. So very hairy on the head, the pronotum, and on the elytra here. Very dense covering, unlike dispar. The only other one that you could confuse it with is dispar, but dispar doesn't have such dense covering of hair, and it has a different shape to the pronotum. And there is another one which we think is probably extinct, and that's Aclypea undata. So these are both vegetarians feeding plants in the beet group, beetroot. This has very rugose punctuation on the elytra there. So I will bring things to a close. If anyone has any more identification questions, don't forget you can contact us via the recording scheme. So here is our page here where you can download the key. You can email us, you can use iRecord, check out our Twitter, or grab yourself a copy of the little picture of the species attracted to light. So I will just go back to sharing my other screen. So what's next? Well, it would be really, really nice to run some more in-person workshops. Being outside and teaching trapping and Silphid ID in the field is way better than doing it online. This is a fantastic medium for now, but it's very engaging to do it outside. Can be a bit smelly, so that's the only bonus that you've got at the moment, is that you're not smelling anything pretty unpleasant. But it's great fun, so hopefully we can do some more of that in the future. I'm also, as I said, quite interested in looking more at the larvae, potentially producing a key for the British species. And another very big project that I've been collaborating on recently is an atlas. This is in collaboration with Steve Lane, who runs the Hysterid recording scheme and is a fantastic British coleopterist, and Colin Lucas, who has been doing all our wonderful distribution maps. So it's an atlas of Hysterids and Silphids of Britain and Ireland, and it's actually finished and it's due out early next year. So watch this space for a copy of that. It's not just an atlas, it is all singing and dancing and has colour plates and keys in to help promote recording of these two families. So it's not a natural pairing, perhaps, in terms of classification, but they are two families that occupy some of the same niches. So both Hysterids and Silphids are attracted to Carrion. So hopefully, if you've already got into Silphids now, then potentially your next group to be interested in will be Hysterids. So yeah, keep your eyes peeled for that. And really all that's left for me to say is a very big thank you. So thanks to everybody that's inspired me and helped me. Thank you especially to Matt and Richard from the recording scheme. Big thanks to Martin Harvey and Jodie and Helen, all at BRC, Darren Mann and Kerry, Max Barkley, Sally Ann Spence. Big thanks to NMS for letting me host today, and to Ed Hill for hosting us in the past. Thanks to Ed, who helped produce our recording scheme logo. And that set to say is keep calm and carry on recording. Thank you all for joining today, and thanks to everyone who's allowed me to use their images in this presentation as well. You can get in touch with us via the scheme or via my email as well, which will circulate if we haven't already with some follow-up resources. So thank you for joining us.
That's great, and thank you, Ashleigh. That was really fascinating for me personally, and certainly lots of positive comments coming through as well. So yeah, I think we've all really enjoyed that, and particularly the microscope view at the end, I thought was excellently done and very visual, very well delivered. So yeah, we could see everything that you were indicating.
I'm glad the tech worked for us.
Yes,
it's a bit touch and go this morning.
Yeah, that was great. So yeah, if there's no other questions just now, lots of positive comments coming through that you'll be able to see yourself, Ashleigh. I will leave it at that for now, and thank everybody for attending and participating in whatever way they did. I'm going to send you a picture after we finished, Ashleigh, and see what you think about this larvae.
Yeah, and same to anybody else, please send any pictures my way. Always happy to put an identification to it if I can.
Great. Okay. Thanks very much,
for joining.
everybody.
Cheers.
Cheers. Bye-bye.